do you know about it?
A molecule has been created that opens the blood-brain barrier on demand. Our brain consists of billions of neurons that must be protected against the influence of unfavorable external factors. The role of this protection is played by the blood-brain barrier. This specialized 650 km barrier between blood vessels and the brain determines what substances can enter the brain. It does its job very well, but from the point of view of neurological diseases it is the worst enemy of modern medicine. It also blocks the access of drugs to the brain. Scientists from Yale University reported in Nature Communications that they have successfully developed a molecule that opens the blood-brain barrier for several hours, allowing the delivery of drugs. This is the first time that a molecule has been used to control the blood-brain barrier, says Professor Anne Eichmann, one of the study's lead authors. Dr. Kevin Bourget joined Professor Eichmann's team in 2017 and began to study the UNC5B molecule. It is an endothelial receptor expressed in capillary endothelial cells. The scientists noticed that the mouse embryos lacking this receptor died quickly because they had not developed a proper blood system. This indicated that UNC5B plays an important role in its formation. Moreover, he found that in such embryos there was a significant decrease in the level of the CLAUDIN5 protein, which is responsible for the tight adhesion of endothelial cells in the blood-brain barrier. The researchers therefore concluded that UNC5B plays an important role in maintaining the blood-brain barrier. It has been known for a long time that the development and functioning of the blood-brain barrier depends on the WNT signaling pathway. Until now, the relationship between UNC5B and this pathway has not been known. Thanks to new research, scientists have noticed that UNC5B acts as a regulator of this pathway. So Bourget went a step further. He deprived the adult mice, with an already developed blood-brain barrier, of the UNC5B receptor and found that when it was absent, the barrier remained open. Next, the scientists decided to check which ligands, molecules that bind to receptors and send signals between and within cells, affect the integrity of the barrier. The barrier turned out to be open when the NETRIN1 ligand is missing. So the scientists developed an antibody that prevented NETRIN1 from attaching to the receptor. After injection of the antibody, the WNT signaling pathway was disrupted and the blood-brain barrier was open for some time. In the near future, scientists want to focus on checking whether this opening of the blood-brain barrier is safe, does not entail any risks, and whether the antibodies themselves are not toxic. This opens the way for more interesting research into how the barrier itself is formed and how it can be manipulated to deliver drugs, says Eichmann.